Hey there, pharmacy students! Today, I'm breaking down the step-by-step process of designing a bioequivalence test for a drug. This is essential for ensuring that a generic drug matches its brand name counterpart in terms of safety and efficacy. So, let's dive right in. First up, we have study design. Bioequivalence studies are usually in vivo pharmacokinetic studies with a crossover design. This means participants receive both the test drug and the reference drug in separate periods, with a washout period in between to clear the first drug from their system. These studies often involve healthy volunteers, although patients with specific conditions can sometimes be included. The sample size, typically ranging from 18 to 36 subjects, is determined through statistical calculations to ensure adequate power. Next, we move on to drug administration. Both the test and reference drugs are administered at the same dose under similar conditions, whether in a fasted or fed state. After the first drug is given, there's a washout period to ensure it's cleared from the body before the second dose is administered. Now, let's talk about blood sampling and analysis. Blood samples are collected at set time intervals, like 0, 1, 2, 4, 6, and 8 hours post-dose, to measure the drug concentration in plasma. Key pharmacokinetic parameters, such as Cmax, the maximum concentration of the drug in the blood, and AUC, area under the concentration time curve, are calculated. These reflect the rate and extent of drug absorption. On to statistical analysis. The FDA requires that the 90% confidence interval for the ratio of the test to reference product for both Cmax and AUC falls within 80% to 125%. This ensures the test drug isn't too strong or too weak compared to the reference. Statistical analysis is generally performed using analysis of variance, ANOVA, on the log-transformed PK data. Finally, we have the FDA submission. Once the study is complete, the full set of results, including all PK data, study design, and statistical analysis results, is compiled and submitted to the FDA. The FDA then reviews the submission to ensure the bioequivalence criteria are met before approving the generic drug for marketing. A couple of key considerations to keep in mind. The study must adhere to good clinical practices, GCP, to ensure ethical and scientific quality, and always follow the FDA's specific guidance documents related to bioequivalent studies for the drug in question, whether it's under fasting or fed conditions, or for modified release forms. This rigorous process ensures that the generic drug performs the same as the brand name drug in terms of safety and efficacy. So there you have it, the step-by-step guide to bioequivalence testing. Stay tuned for more tips and insights on your journey through pharmacy school. Catch you next time.